Hello everyone. Today I'm presenting a review by academician Yuming Zhang's team, submitted to iMetaOmics, titled Strategies and Mechanisms of Precision Genome Engineering. We will explore the evolution, core mechanisms, and future of genome engineering. The field of genome engineering is experiencing a profound paradigm shift. We are moving from random, cleavage-based gene editing to precise, predictable genome writing. This transforms the genome into a programmable biological information platform. This transition highlights four key advancements: deterministic over random editing, reduced double-strand break genotoxicity, RNA-guided payload expansion, and AI/structural biology-driven de novo editor design. Collectively, they usher in generative biology. Historically, genome engineering spans three stages: nuclease cleavage, precision editing, and genome writing. Early tools like CRISPR-Cas9 simplified targeted cleavage. Next, base and prime editing reduced reliance on double-strand breaks. Today, systems enable large-scale engineering, shifting our goal from simply cutting genes to writing genomes. This journey started with CRISPR nucleases like Cas9 and Cas12a, which create double-strand breaks. However, repair outcomes depend on unpredictable cellular pathways. Despite optimization efforts, these breaks can still cause severe risks, including large deletions, chromosomal translocations, unintended cellular selection pressures. To mitigate these risks, the field developed precision tools. Base editors fuse a Cas9 nickase with a deaminase to perform specific C to T or A to G conversions without breaking the DNA double strand. Recent engineered variants have significantly improved efficiency, narrowed the editing window, and reduced off-target effects. For more complex rewrites beyond simple base conversions, we use prime editing. It uses a reverse transcriptase and a pegRNA that carries new genetic information, synthesizing the desired sequence directly into the genome. Upgrades like PE3 to PE5 further improve efficiency by optimizing cellular mismatch repair. Precision engineering also extends beyond DNA sequences to regulate gene expression. Tools like CRISPR-I, CRISPR-A, and Cas9 fusions allow us to reversibly activate, repress, or modify chromatin states. We can now reconstruct 3D genome architectures or precisely regulate RNAs, all without altering the underlying DNA sequence. Because early tools lacked payload capacity, large-scale genome writing technologies emerged. Systems like Cas use RNA-guided transposition for multi-kilobase integration. PASTE combines prime editing with integrases for massive payloads, while Bridge RNA introduces a novel mechanism for complex DNA insertions and inversions without double-strand breaks. With programmable writing, the genome can now function as a biological recorder. Tools like Camera and Ngram use base or prime editing to log the intensity, duration, and order of cellular events directly into DNA. Complex editing relies on AI and delivery innovations. AI protein language models now design more compact, less immunogenic editors from scratch for delivery alongside viral vectors. Non-viral methods like lipid nanoparticles, LNPs, and engineered virus-like particles, EVLPs, offer safer, transient, and more controllable options. Delivering the editor is just step one. Ensuring safety requires multidimensional validation. We've moved from checking local sites to whole genome evaluations. Techniques like guide-seq map off targets, long-read sequencing reveals structural abnormalities, and perturb-seq link specific edits to single-cell phenotypes, ensuring systemic safety. Despite these leaps, challenges remain. It is still difficult to simultaneously optimize efficiency, precision, and payload capacity. Issues like product impurity and in vivo delivery hinder clinical translation. Solving this requires combining AI, structural biology, and novel delivery platforms eat safer, transient synthetic editors. In conclusion, genome engineering is shifting from random, break-reliant editing to precise, large-scale writing. Supported by AI, synthetic biology, and new delivery tech, we are overcoming current limitations. Ultimately, the genome is evolving into a truly programmable, writable, and dynamic biological information platform. Thank you.